Okay, are there any questions? You should be seeing my screen now. Not much to see. There it is. Syllabus. All right, not hearing any questions. Can somebody please confirm you can hear me?
I can hear you. Confirming.
All right, today is February 10th, and we should be doing Lab 7, throat cultures and urine cultures. And tonight you should get your first unknown results and your unknown number. Any question about any of that? All right, Lab 7, throat and urine cultures. There is some reading, as is normal. Oops, too far. So please read page 392 about nose, throat, and mouth normal microbiota. Read about beta hemolytic streptococci. You can find that reading at the end of this lab module in the appendix. You can read about Haemophilus also in the appendix. Read about the oxidase test on page 132 of the textbook. Read about the catalase test on page 155. And read about the coagulase production test also in the appendix of this lab module. And I added something to the appendix in the lab module to discuss the coagulase and the Haemophilus, the beta hemolytic streptococci. So I'll added that to the lab module. Any question about any of that?
Not on any of that. I do have a question for the unknown project.
Uh-huh.
Can we expect the email to come out today, or is it going to be tomorrow for the results for the first gram test?
It should be sometime tonight, but it might be after midnight. So technically that's tomorrow, but
No
I
worries.
send it out tonight. Okay. I will send it out tonight. Um, I usually don't send it out until at least 10 o'clock. Um, and I doubt I'll get it out earlier tonight. And sometimes I get it out after midnight. So it might be between midnight and 2 a.m., but it will get out tonight. Okay.
Okay,
Oh, good.
wonderful. Thank you.
My computer's going to speed up. It just finished the lecture. All right, let's get out of here. The lecture file just got made. And where was I? Here we are. All right, this lab will have some video clips. We no longer have a virtual lab for this lab. Upon successful completion of this lab, you should be able to one, explain how to properly collect throat and urine specimens for routine culture, two, list common bacterial pathogens isolated from throat and urine cultures, three, know the culture media is used to cultivate these pathogens, four, recognize which tests are used to identify throat and urine pathogens, and five, understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. So you need to understand them and be able to explain them. We will briefly talk about the XV factor testing, but you don't, I'm not going to ask you any quiz questions about it, so you don't really need to know about that one. Any question about any of that? All right. So, in an introduction, once a specimen is received in a lab, it'll be logged and processed in a timely fashion. Culture media will then be selected to inoculate it and then incubated. And that's what happens to a specimen collected from a patient that is sent to a lab to isolate and study the culture. For throat and urine specimens will be what we're looking at today. Below is an overview of culture media used to cultivate common bacterial pathogens isolated from the throat and the urine. So in throat, we have one main agar, blood agar, which is our culture media for most things, especially in adult. In a child, we also inoculate chocolate agar as well as blood agar. Streptococcus pyogenes is the main culprit found in the throat of all adults. It's only in children where we find both Streptococcus pyogenes and Haemophilus influenzae. And usually it's young children or infants that have Haemophilus influenzae. To grow Haemophilus influenzae, it will not grow on blood agar, but it will grow on chocolate agar. Streptococcus pyogenes is a fastidious organism. It will not grow on a general purpose media like Triptychs soy agar, but it will grow on blood agar. Haemophilus influenzae is also fastidious, but it will not grow on blood agar. It has to grow on chocolate agar. Any question about any of that? All right, for urine specimens, we always grow them on two media. We always grow it on blood agar, and then we also grow it on either McConkey's agar or EMB. Blood agar can grow everything that we're looking for in the urine specimen. So that's why we use blood agar, meaning gram positive as well as gram negative. But most of the cultures we get from urine will be a gram negative. So we grow them on either McConkey's agar or EMB agar to select 4 gram negatives. And so if you have a urine specimen, you always grow it on 2 different media, blood agar and then either McConkey's agar or EMB. For Streptococcus species, Enterococcus, Staphylococcus, they are gram positive and they will grow on blood agar. However, the number one organism we find in urine cultures is actually E. coli. And E. coli and other members of the Enterobacteriaceae family, which E. coli is a member of, will grow on blood agar, but we tend to grow it on either McConkey's or EMB agar because E. coli or Enterobacteriaceae will grow on these 2 media, and it'll select against gram positives. Pseudomonas, especially Pseudomonas aeruginosa, is another gram negative that is frequently found in urine cultures, as well as other members of Enterobacteriaceae. So there are a number of species that can contaminate urine and can be isolated from urine, but there's really only 2 you need to know about that come from the throat, and only 1 if you're an adult. If you're a child, especially a small child, there are 2. Any question about that? All right, throat cultures. Sore throats caused by a bacterial infection usually has a white patch as well as red patches. If you only see red patches, that sore throat is likely viral, but if it has white patches as well as red patches, it's likely bacteria. The most common bacterial pathogen isolated from the throat is Streptococcus pyogenes. To get a throat culture to isolate a bacterial pathogen, meaning Streptococcus pyogenes, you always want to streak the back of the throat and the tonsils. You do not want to streak the sides of the mouth, the tongue, and not shown the top of the mouth. And a tongue suppressor will help you get that. Any question about any of that? You then run different tests to isolate it that is Streptococcus pyogenes. Patients that test positive for Streptococcus pyogenes are almost always put on antibiotic therapy. The reason is Streptococcus pyogenes has a cell capsule, and that makes it very difficult for the host to throw off the bacterial infection. Streptococcus pyogenes is fastidious and it will grow on a blood agar plate. So we inoculate a blood agar plate and then take a look at the colonies growing on the blood agar. We're looking for small white colonies showing beta hemolysis around the colonies. Because Streptococcus pyogenes takes on that characteristic, showing small white colonies with beta hemolysis. Once you have small white colonies with beta hemolysis, you only need to run 2 tests to confirm that it's beta hemolysis. Excuse me, that it's Streptococcus pyogenes. You have to run the catalase test and the basotrace and susceptibility test. So here we've done a throat culture and one here and one here. Let me blow that up so you can see it here. This patient is pretty much a normal throat culture where you can see many of the colonies have no hemolysis, like that one there. That one has alpha hemolysis, a dark ring around it. And then there's some which are very weak, either alpha or gamma hemolysis. I'm not sure how to read those, but that one's definitely gamma. But that one there, I'm not sure about. That one is definitely alpha hemolysis. And there's some alpha in there. I can see the darkening there. But like I said, there's a lot like that one there, probably that one there. Look gamma. And normally in the throat, we have many bacteria. And some of them are gamma, some of them are alpha, but none of them in the normal throat culture contain beta. And here we have a person with beta hemolysis from the throat, and there it's kind of blurry, but there's the colony, the small white colony in front, and you can see beta hemolysis showing the clear region of the blood around the colony. Any question about any of that? And there you can see alpha hemolysis, but there's beta, there's beta, there's beta, and there's beta, and there's some beta in there. There is some alpha in there, like right there and right there. And that actually looks like that could be a gamma right there. Once you have beta hemolysis on the blood agar, you then run the catalase test. Catalase test, if the bacteria make catalase, they will take hydrogen peroxide and make water and oxygen, and the oxygen will appear like bubbling. See if I have a picture of that. There we go. There's catalase negative. This bacteria is producing catalase. You can see the bubbling from the oxygen being generated. The oxygen comes and the water comes from the breakdown of hydrogen peroxide by catalase. If bacteria taste positive for catalase production, bubbles are observed, and the bacteria is not Streptococcus, because Streptococcus and Enterococcus are 2 families of bacteria which are catalase negative. They can grow in air, but they are catalase negative. Most bacteria that grow in air like Staphylococcus, are catalase positive, but Streptococcus and Enterococcus families are catalase negative. And then you need to do one more test to show it's Streptococcus pyogenes. So if you have beta hemolysis on blood agar and catalase negative, you then need to do the bacitracin test. What you do is you smear the bacteria over a region of the dish, put bacitracin antibiotic disc on the plate, and if you have a zone of inhibition around the disc greater than, I think, 14 millimeters, then that is sensitive to bacitracin, and that would be Streptococcus pyogenes, which is the same as group A Streptococcus. If, on the other hand, it is bacitracin negative, either no hemolysis or hemolysis less than 14 millimeters, then that's catalase negative, but it's bacitracin negative. It's not resistant to bacitracin. So it's, I mean, not sensitive to bacitracin, sorry, is resistant to bacitracin. And that would not be Streptococcus pyogenes. And those are the only two tests you need to do for a beta hemolytic bacteria. I'll warn you, in the lab, it made the assumption that it was already family Streptococcus, beta hemolysis, and then you don't need to do the catalase test. All you need to do is the bacitracin test, because it's already known to be Streptococcus. And anyways, if it's bacitracin sensitive, catalase negative, and beta hemolytic, then it is Streptococcus pyogenes. Another organism we can get in the throat, especially in young children or infants, is Haemophilus influenzae. Haemophilus influenzae is a gram-negative coccobacillus. It's a common cause of ear, eye, and sinus infections in babies and young children. Haemophilus influenzae is fastidious, but it will not grow on blood agar because it cannot lyse the red blood cells itself to release the factors that it needs, NAD or V factor, and hemin or X factor, which, by the way, Streptococcus pyogenes does need, but it can lyse the red blood cells. So that's why it can grow on blood agar. Haemophilus influenzae can grow on chocolate agar because chocolate agar is the same as blood agar. It's just we add the blood when the agar is very hot, and that lyses the blood, making it look chocolate in color. And there you can see Haemophilus influenzae growing on a chocolate agar. You could get Haemophilus influenzae to grow on blood agar if you streak Staphylococcus aureus on the blood agar. So here we have a blood agar plate, and this streak here is a single streak of Staphylococcus aureus, which you can't really see. Now you can see a little bit. That streak there down the center is the streak of bacteria. The whiteness is from the lysis of the red blood cells with the Staphylococcus aureus. And then we streak this way, Haemophilus influenza, on the plate perpendicular to the blood agar. And you can see that the Haemophilus influenzae can grow in this region of the plate. It cannot grow in this region of the plate. The reason it doesn't grow here is because Haemophilus influenzae must receive nutrients from the blood cells, and Haemophilus influenzae does not lyse the blood cells, so Haemophilus influenzae cannot grow. However, Haemophilus influenzae can grow in this region of the plate because Staphylococcus aureus is lysing the blood cells right here, and then the nutrients from the blood can diffuse to this region, and that's why Haemophilus influenzae can grow near the Staphylococcus aureus. Any question about any of that? Hopefully yours will be showing better than that. I'm not saying what that is. There's the Staphylococcus aureus streak, and there's the Haemophilus influenzae growing near the Staphylococcus aureus. All right, any questions about Haemophilus influenzae or Streptococcus pyogenes? Two bacteria you can isolate from the throat of a patient. Haemophilus influenzae usually from young children or infants. No question. Let's move on to urine cultures. Urine cultures are used to diagnose urinary tract infection. Bacteria can travel from the urethra to the bladder, causing a urinary tract infection. They can also travel from the bladder to the ureter, and then from the ureter to the kidney. My mother actually had an infection all the way up from her urethra up to her kidneys. You don't want that, by the way. That's a very serious infection. You want to get it early, either in the urethra or no further than the bladder. Females are more predisposed than males to getting a UTI, urinary tract infection, because they simply have a shorter urethra. How you obtain a urine culture is usually you clean the genital area prior to collection, and then you catch midstream in the flow of urine, in the middle of the stream. And that way you reduce the number of contaminants in the urine. If you catch, like I said, only like the beginning urine or the end urine, you'll get more contamination. However, even midstream, you get a lot of contaminants from the urethra. It would be better to catheterize the patient, sticking a catheter up the urethra and then isolating the bladder directly. But as you can see, you have to stick the catheter up the urethra, and the urethra does have bacteria on it, and you could put the bacteria from the urethra into the bladder and then sample that. So the catheter is not perfect either. You do get contamination from the catheter. The best way to do it is the suprapubic aspiration, where you put a needle through the abdominal wall. You sterilize the abdominal wall, put the needle through the abdominal wall into the bladder, and then sample the urine directly from the bladder. That's the best way to do it. It's also the most invasive and the way that patients would least enjoy, or they'd be the one they'd least object to, I guess would be a one would say. So they most likely would like the clean catch midstream. They'd be more likely to take the catheter and least likely to take the suprapubic aspiration. Any question about any of that? But the suprapubic aspiration is the best way to get a urine sample. Least contamination. Once you get a specimen of urine, you point 1 microliter or 0.001 milliliter of urine onto a plate, such as blood agar, and then streak that over the plate. And let me show you this right here. That measures out 1 microliter of urine, or 0.001 milliliter of urine, and then you streak it over the plate. Usually go like that and then streak that way. Take a look at what grows, and then you take your urine specimen and then streak it out on McConkey's or EMB plate specifically to grow gram negatives because most urinary tract infections are gram negatives. The number one will be E. coli. It's a little less than 50%, but it's like over 40% of urinary tract infections are caused by E. coli. The interpretation of a positive urine culture is multifaceted. Factors to be considered is the type of collection. The clean catch midstream will be the most contaminated, catheter less contaminated, suprapubic aspiration, least chance of contamination. The gender of the patient, males have less contamination than females. The number of colonies per mil, you consider that. The presence of polymicrobial growth. If you had multiple organisms isolated, it may be a poor indication, a poor collection technique. The patient may have not collected the urine with clean midstream. They either did a poor job of cleaning off their genital and then collecting it, or they may have not waited to the midstream of the urine to collect it. Any question about any of that? All right. Once you get something growing on blood auger, it always will grow on blood auger, as well as possibly on MAC or EMB, you then run some further tests to isolate the culture. Some tests we're going to talk about is the coagulase test. I'll just mention the test we're going to talk about. The coagulase test, the oxidase test, and the catalase test. We already mentioned that one. Coagulase test, if the bacteria can produce coagulase, it will create clumping or a clot in the sample. And Staphylococcus aureus is the main bacteria which produces coagulase. And if you find coagulase happening, it's probably from Staphylococcus aureus. Coagulase is important because it can contribute to the pathogenicity of Staphylococcus aureus. What happens is the fibrinogen with the presence of coagulase will create a fibrin clot. So we'll see the plasma start to clot on a slide, coagulase negative, or you can do the test tube, and the clotting will cause a clumping of the plasma. If it's coagulase negative, there will be no clumping of the plasma. It's thought that the fibrin clot can help protect the Staphylococcus aureus from the host immune system, that the host immune system will have a hard time getting through the clot and then attacking the Staphylococcus aureus. So that a bacteria like Staphylococcus aureus that produces coagulase is better at resisting the immune system than other bacteria that do not produce coagulase. And if you know the bacteria is a member of the Staphylococcus family, and then you do the coagulase test, if it's coagulase positive, Staphylococcus aureus is the only bacteria in the member of the family of Staphylococcus that produces coagulase. Any question about any of that? All right, the most common bacteria isolated from the urine is E. coli. E. coli is a member of the family Enterobacteriaceae. Pseudomonas aeruginosa is a member of the Pseudomonaceae family, so a separate family than Enterobacteriaceae. People like to determine is the gram-negative infection in the Pseudomonaceae family or the Enterobacteriaceae family. And usually the test that they use to separate them is the oxidase test. If the bacteria is in the Pseudomonaceae family, like the pathogen Pseudomonas aeruginosa, it will be oxidase positive. But if it is in the Enterobacteriaceae family, it will be oxidase negative. So the oxidase test is the test we use to distinguish if it is gram negative, if it's in the Pseudomonaceae family or in the Enterobacteriaceae family. There are other tests you can use, but the oxidase test is a test that can be done immediate, and then you can get the results, well, in a few minutes. Other tests take time, like the glucose fermentation test. You could do that one. I'm forgetting the other test you could do. Glucose fermentation. All members of the family Enterobacteriaceae are glucose fermentation positive. All members of the Pseudomonaceae family are glucose fermentation negative. The other test you can run is the oxygen requirement. Pseudomonaceae family or require oxygen presence. Enterobacteriaceae family are facultative anaerobes, so they do not require oxygen. Any question about any of that? So you could use all three of those tests, but usually the oxidase test is the one done because it can be done immediate. If you have the cells around, it can be done immediate. They don't have to be fresh cells, but it helps if they're fresh cells. And it can also, you get the results right away. Oxidase is looking at the production of oxidase C, cytochrome C oxidase, or cytochrome oxidase C, and all members of the Pseudomonaceae family have that enzyme, so they're oxidase positive. If the bacteria are oxidase positive, they'll turn blue or purple in the reagent. So this would be the reagent, and there's the bacteria there, and that's oxidase positive. If it is oxidase negative, there will be no change in the plate. So this would be this region here is oxidase negative. We probably have no cells there, by the way, but this is oxidase negative. All right, any question about the oxidase test or the coagulase test or the catalase test? If not, you're not going to be physically performing any of these activity. Everything we do will be done online. However, you will be held responsible for knowing how to perform and do them. We have some video clips made by Dr. Gary Geyser. So we have a little video clip on the catalase test. We have a little video clip on the coagulase test and a little video on the oxidase test. So watch these videos and then do your lab. Let's go to the worksheet. Answer 1. So for questions 1 to 3, discuss whether you're using the enzyme catalase, coagulase, or oxidase. So in 1A, which enzyme converts fibrinogen into fibrin? And then 1B, name a microbe that tests positive for this enzyme. For 2A, name the enzyme that detects the presence of cytochrome C proteins, cytochrome C oxidase in the electron transport system. And then 2B, name 2 microbes that test negative for this enzyme. And you can state a species or a family here, or even the genus here. For 3A, state the enzyme that hydrolyzes hydrogen peroxide into non-toxic products, water and oxygen. And then for 3B, name 2 microbes that test negative for this enzyme. Once again, you can put the family name or the species name for 3B. For question 4, which enzyme is used to differentiate between the following? So which enzyme differentiates between Staphylococcus and Streptococcus? These are 2 different families. And then which enzyme differentiates between Staphylococcus aureus from other members of the Staphylococcus family? And then 4C, which enzyme differentiates between Enterobacteriaceae such as E. coli and Pseudomonaceae? Question 5, concerning specimen collection for routine throat culture, what areas of the throat should be swabbed? 5B, what areas of the mouth should be avoided? 6. Oh, by the way, for A and B, you need to have at least 2 answers for each. For 6, what is the most common species causing a bacterial throat infection? So don't say something like the common cold, because it's not a bacterial throat infection. 7. What test is used by microbiologists to differentiate between beta hemolytic group A Streptococcus, being Streptococcus pyogenes, from 8 other beta hemolytic Streptococcus species isolated in throat cultures? So answer that test there. 8A, state 2 non-selective agar plates that are inoculated following a child's throat culture. And you must state the culture, because if you give another culture, you have to add additives like a general purpose media. You need to add an additive, which I will accept that answer, but you have to state the additive, and it's better just to state the media that supplies the additive. And 8B, what organism can be isolated on CAP but not on PAP? Question 9, list 2 media that are used to grow urine cultures. Question 10, list 3 reasons why a urine specimen should be rejected for culture, and you can find that in the appendix of the lab module. 11, how much urine is placed on the surface of a blood agar plate? You can tell me either in microliters or in milliliters. Don't give me the answer in, I don't know, like liters, for example, because I don't know off the top of my head and I don't want to calculate it. So give it to me in microliters or in milliliters. 11. All right, 12. What is the most common bacteria isolated from patients with bladder infections? You need to name the species. And then 13, identify the Gram reaction, give the Gram stain, and the cell morphology, the shape, not the growth arrangement of the bacteria. You can find the answer in the module, but you may need to do some research, like you might need to look in the appendix. Pseudomonas aeruginosa, list the Gram stain, and then list the cell shape. For E. coli, list the Gram stain, list the shape. Staphylococcus aureus, list the Gram stain, list the shape. Streptococcus pyogenes, give the Gram stain, give the shape. For Haemophilus influenzae, list the Gram stain, and then list the shape. I'll let you know with Haemophilus influenzae, it needs to be one of 2 answers. I will accept either or one of the 2 for something, for its shape. I'll say that. All right, any questions about any of this? It's 7:18. I will be here until 8 o'clock to help you with the lab if you have any questions. But if you have a question, make sure you ask before the last student leaves, because once the last student leaves, I'm going to log out as well. All right, any questions?